Hi everyone, let's get started. Today we're going to be discussing microbial metabolism, part 1. The first thing we need to do when discussing metabolism is to give a definition to this term. When we use the word metabolism, we're talking about all of the chemical processes that occur within a microbial cell that are required to maintain the life of that cell. If we look at all the various metabolic processes that occur in all cells, we can break them down into two really big groups. The kinds of processes that break things down and the kinds of processes that build things up. We give these two groups names. We talk about anabolic reactions and catabolic reactions. Any metabolic reaction that involves the building up of new molecules is an anabolic reaction. And all anabolic reactions require energy. So we also refer to them as being endergonic. If you break down that word endergonic, the ender part means into or within. So anabolic reactions, the kinds of metabolic reactions that build things, build molecules, are going to consume energy. In comparison, any metabolic reaction that breaks something down, breaks something apart, is a catabolic reaction. And catabolic reactions are going to release energy. Remember, if you break a bond, the energy that's in the pair of electrons that are sitting in that bond are going to be released. So that's an exergonic reaction. If you recall from early on in the course, one of the first things we talked about regarding microbes is how amazingly diverse they are. And when it comes to metabolism, we see this same kind of just remarkable diversity. One of the things that really distinguishes microbes, bacteria and archaea, and single-celled eukaryotic organisms is their amazing array of metabolic processes. They're capable as a group of a whole lot more different types of metabolism than you and I are. So in order to get a hold on all of this diversity, one of the things that we do is we categorize microbes into what we call energy classes. And we're going to describe them based upon two very basic things. Number one, where they're getting their carbon from. Remember, if you live on planet Earth, you're building molecules that contain carbon for the most part, a carbon skeleton, if you will. And that's one of our categories of energy class. But the other one we use is how microbes obtain their energy to do all of this building. On this next slide, we can see a diagram that helps us classify microbes into these two groups, where they obtain their carbon and where they obtain their energy. So when it comes to energy in the microbial world, cells are either getting energy from chemicals, from molecules, specifically the covalent bonds inside of molecules, or they're getting their energy from light. Remember, there are photosynthetic microbes, and just like our photosynthetic plants, these organisms are capable of harvesting energy, collecting energy from sunlight. So if you're a microbe that gets its energy from a chemical, from a molecule, or I should say a series of molecules, we refer to you as being a chemotroph. The process of getting energy out of a chemical is called chemotrophy. Now, if you're in the other category and you're an organism that's capable of getting energy from sunlight, you are a phototroph. And the process that you're using is called phototrophy. Now, if you look down under chemotrophy on our diagram, you can see there are actually two kinds of chemotrophy that occur. There are the chemotrophs that get their energy out of organic chemicals. And remember, in scientific terms, organic means carbon containing. And then there are chemotrophs who are capable of getting energy out of inorganic molecules, molecules that don't contain carbon. So for the organic group, we're going to call them chemotrophs, but more specifically, we're going to call them chemoorganotrophs. Break that word down to mean an organism that gets its energy from organic chemicals or molecules. In comparison, a chemolithotroph is an organism that gets its energy from inorganic chemicals. These organisms, the chemoorganotrophs and the chemolithotrophs, are using chemicals, and specifically the covalent bonds inside chemicals, what holds those molecules together. They're breaking apart those covalent bonds in order to get energy so that they can build ATP. Now, over on the side of the phototrophs and phototrophy, these we don't break down any further. Any organism that's capable of phototrophy, or what we would call a phototroph, is harvesting energy from sunlight and using that energy to build ATP. So, catabolic reactions are metabolic reactions that break things apart and essentially release energy. Catabolic reactions involve a breaking apart of the covalent bonds inside molecules. Anabolic reactions, on the other hand, require energy because anabolic reactions build new covalent bonds, build new molecules. So inside of any microbial cell, there are lots of different catabolic reactions going on and lots of different anabolic reactions going on. But among all those different types of reactions, we can say something that applies to almost all of them, and that is that they require enzymes. Most biologically important reactions, metabolic reactions, require an enzyme in order to occur. All enzymes are catalytic proteins. They're proteins that are capable of doing this job called catabolism. And all that means is they help reactions proceed. They help reactions occur more readily, more easily than they would otherwise. Enzymes will actually increase the rate of these biological reactions, and that's important if you're a living cell. Metabolism has to happen quickly. Organisms have a need for things in real time, and so enzymes allow these reactions to happen with real speed. The other thing to know about enzymes, and all catalysts in fact, are that they're not used up in the reaction. Enzymes get used over and over and over again. And we can identify these critically important molecules just by looking at their name. I've mentioned to you before that if you look at the name of a molecule and it has an ASE at the end, you know that you're looking at the name of an enzyme. So things like amylase and peptidase and catalase, these are all enzymes. So enzymes are just proteins that happen to be capable of this thing called catalysis. They're capable of catalyzing metabolic reactions. And like all proteins, enzymes are made out of amino acids. In other words, amino acids are the subunits, the building blocks of proteins. We take one amino acid and we join it to another amino acid with a very special kind of covalent bond called a peptide bond. And what happens is a long chain of amino acids gets built. And then that long chain simply starts to fold. Each of the R groups in each of the amino acids, remember we talked about those, they're going to be attracted to other R groups or they'll be repelled by R groups. We'll start to see hydrogen bonding and van der Waals interactions and other kinds of weak attractions occurring among the amino acids in this long chain. And it won't remain as a long chain. It will fold up into a very intricately shaped three-dimensional molecule. Every protein has its own unique three-dimensional shape, and it's the shape of that protein that determines its function. So if you alter the shape at all, you're going to alter the function. Enzymes are no different. Each enzyme has its own unique three-dimensional shape, and it's its shape that allows it to catalyze a particular metabolic reaction. In the picture down at the bottom of this slide, we're simply looking at this folding process. Over on the left-hand side, they've drawn a long chain of these blue circles. Each one of the blue circles represents an amino acid, one of the 20 types of amino acids that proteins are made from. And then you can see after that chain has been made, it begins to fold. It folds up into structures called alpha helices and beta pleated sheets. And then those alpha helices and beta pleated sheets start folding up and interacting with each other until ultimately we have this very complex three-dimensional folded protein. Sometimes these three-dimensional folded proteins work together to make an even more complex molecule called a quaternary protein. Don't worry too much about that term though. Just understand that it's the three-dimensional shape of proteins like enzymes that allow those molecules to do the important work that they do. So every enzyme is specific to a particular metabolic reaction. Enzymes have locations on them that are structured in a way to allow substrate molecules to bind. We refer to these very specific locations as active sites. So remember, all a substrate is a reactant, a molecule that's going to come into a metabolic reaction, go through some kind of change, and become product. So that's all I mean when I use the word substrate. It's the molecule that's coming in to a reaction and it's going to be transformed into some kind of a product molecule. And these substrates bind to the enzyme at a very particular location called an active site. Remember, enzymes are really interesting proteins because they are used over and over and over again. They're not single-use items, in other words. They're capable of doing this work in a cell for a relatively long period of time. If you look down in the diagram at the bottom of the slide, you can see an enzyme depicted by this sort of gold-colored orangey molecule. And you can see that there's a very deliberate wedge missing from the circle. The artist is trying to show us that this empty area right here, this wedge-shaped empty area, represents the active site. Up here, this very bright yellow molecule represents the substrate. And you can see that the substrate binds perfectly with the active site. The two things are shaped so that they fit together perfectly. This is what allows enzymes to be so specific for certain reactions. Only this substrate is going to fit this well into this active site. Now, once the substrate is bound to the enzyme, the catalysis occurs. The enzyme is going to help the metabolic reaction proceed. And that's what they're showing us over here. The substrate molecule has gone through a change. It's become product molecule. And in this diagram, that's represented as this bright blue circle and this green sort of triangle molecule. Once the substrate becomes product, the product is going to be shaped just a little bit differently than the substrate is. So the product molecules don't fit as nicely in the active site, and they will thus get released. It's the release of the product from the enzyme that allows the enzyme to go back to its original shape. And now this enzyme is ready to do the same thing all over again. It can bind to the yellow substrate. It can catalyze the reaction, help the reaction proceed. We'll get product formed. Then the product gets released and the enzyme goes back to its original shape. This happens over and over and over again in living cells. In this diagram, we can see another artist's representation of this process. I like this diagram because the enzyme is shown in relation to the substrate on a more realistic scale. Enzymes are proteins, and proteins can be enormous. Proteins can have hundreds and even thousands of amino acids in them. In this drawing, each one of these green circles, these tiny green circles, represents a different amino acid. This is a big, big molecule. And again, you can see there's this very obvious location on this molecule that we would call the active site, the place where the substrate, in this case glucose, is going to come in and bind. And glucose fits perfectly in this active site. When the glucose comes in and binds here, the binding action is going to cause the whole enzyme to change shape a little bit. And that's what they're showing us over here. You'll notice that the enzyme has kind of closed in around that substrate. And that's because the substrate causes the enzyme to fold a little differently. There are different hydrogen bonds formed and different van der Waals interactions going on when you attach that substrate to that enzyme. It becomes basically a brand new shape once the substrate is bound. This shape change is going to allow catalysis to occur. It's going to allow product to be formed and then released from the enzyme.